Welcome, everyone. My name is Benjamin King, and as part of the main INBRI program, we have developed a set of training materials to help students and researchers leverage cloud-based computing environments for bioinformatics analysis. And in this video, we will be reviewing a tutorial that we have made on analyzing a bacterial RNA sequencing study. And as part of this, we will be featuring the Google Cloud Platform as a commercial computing environment. There are a set of 3 tutorials that we will review, 2 of them in additional videos. And this video is going to be featuring tutorial 1, where we will be analyzing a subset of data from a bacterial RNA sequencing study. We're using a subset so that the analyses take very little computing resources. Separately, there's a tutorial that analyzes the complete data set, and that is featured separately. And the output of this tutorial is going to be a set of read counts. And these read counts can then be analyzed in R using the RNA-Seq package, and that's featured in a separate video. And the output of that third video is the set of differentially expressed genes. So this gives you an orientation, and again, we're starting here with tutorial 1. So to access and use these training materials, what we'll be doing is to first go to the GitHub resource so that I can show you the written training materials that we've developed, and also tell you a little bit about the IPython notebooks that we've developed. Then we'll be visiting the Google Cloud Platform, where I will be logged in to GCP. And then we will launch a virtual machine that allows us to run the notebook. So again, following this link, we can access GitHub. And at GitHub, we've made a set of training materials and which compose currently of these 4 tutorials. And this diagram just gives you an overview of what these tutorials are and a brief description, different requirements, a little bit about the data that we're analyzing from Sally Malloy's laboratory here at the University of Maine. And importantly, there are steps that we briefly describe where of how to take the notebooks that we've created as part of this tutorial and to import them into GCP and then to execute them. So these are step-by-step written instructions on how to do that, and but I'll be demonstrating that for you. So I'm going to scroll back up to the top and tell you a little bit about the structure of what we've made available here in GitHub. First, there are a set of IPython notebooks. And these IPython notebooks are documents where every step can be written separately. So I'm going to look at this notebook here, which is the focus of this tutorial. So I'm clicking that link, and this is what that IPython notebook looks like. It first begins with the analysis workflow that's listed. We're starting with our FASTQ files. We're trimming them. We're doing some diagnostic analyses as well. Then we're aligning those trimmed sequences to a reference transcriptome of the bacteria, M. chelonae. And then we're generating read counts for every sample. And in this notebook, all of the steps are clearly described. And one thing that's important to note, and is a major obstacle for many people getting started in using these technologies, is that all of the software that you use must be installed on some server. And that is sometimes quite difficult to do. But what we'll be using is a technology called Mamba Forge. And Mamba is a way of installing packages very easily on different computing environments. And you'll see some of the names of the programs like Trim-O-Matic or FastQC, another one called Salmon. So those are featured here in this workflow. And once you set up Mamba, then it becomes very easy to install these packages. And so we'll be running each of these steps in the workflow separately so that you can see how all of the steps are run. And this goes through the entire workflow. This notebook is something that we're going to run on Google Cloud Platform. However, this notebook could be run on any server, whether that be your on-premise high-performance computing system or some other server that you have in your lab. But it can also be used on any commercial cloud computing platform. So these notebooks are reusable. But we'll be featuring specifically Google Cloud Platform. So back a page to the home page and scroll down to the Getting Started section. There are a set of instructions that walk you through the process of using specifically Google Cloud Platform to set up this virtual machine and then import the notebooks. And then I'll show you all of these steps. So what I'm going to do next is to log into GCP. To log into the Google Cloud Platform, one goes to the address cloud.google.com. And once you're logged in, you can click this console link here in the upper right-hand side. That will then log you in, and your account will be attached to a project. And your project will be different than mine, and those have to be tied towards some kind of a billing account. The first step that we're going to do is to start by launching what GCP calls a VertexAI workbench. So under the navigation menu, some people call the hamburger menu, we'll click this, and we have to scroll down. There are many different services that they provide at GCP, and I'm scrolling down until I get to the AI or artificial intelligence section. So I'm scrolling more. There we are, artificial intelligence. And the VertexAI section here of the menu, we're going to click on Workbench here. So I'm going to execute this. This allows us to run IPython notebooks. In this area, I've run previously a number of different notebooks. And so what I'm going to do right now is to start a new notebook that's going to be specific for this tutorial. So I'm going to click on New Notebook. And I can launch a new notebook using, in this case, we want to use Python. There is another tutorial that we'll do where we'll be running things in R, but we're going to choose Python 3 here. You have to give it a name. And so I'll just say video demo. When you set up a virtual machine, you can choose the region Google Cloud Platform has many data centers around the world, and I'll use the one in Northern Virginia as it's closest to us. And then the zone, I'll just choose C. Then we're going to choose the default here in terms of the configuration of this virtual machine. So it has 4 virtual CPUs and 15 gigabytes of RAM with 100 gigabytes of disk. We might, depending on your analysis and things, one might need to change these configurations, and you do that using the advanced option. So if you needed more disk space or the like, one could configure that there. And finally, we're going to click Create here to create this notebook. And it will be, so video demo now is in our list of servers, and it is currently creating this environment. When this is done, there's going to be an Open Jupyter Lab link that we can click, and that will be here momentarily. So now the link is available, and we're going to click Open Jupyter Lab. And then we get the Jupyter Lab interface, which looks very familiar to other when you're running Jupyter Lab on other servers. So this is sort of the launcher page. And what we need to do next is to retrieve the IPython notebooks from GitHub. And we're going to do that under the Get menu. And we're going to clone a repository. So that's a way of copying the IPython notebooks from the GitHub repository that we've made. So get clone a repository. Then I need to enter the URL. This URL is on the training materials site. So I'm going to just show you that it's right here, this URL here. So, and then I'll click clone. And so now it's cloned the repository, and I'm going to click on this folder icon here on the left so that we can look at the contents. And we now have a set of notebooks, IPython notebooks. And the one that we'll be using is this one here, the short demo. And I'm double clicked on this, and now we can see the IPython notebook. So this looks very similar to what we saw when we were in GitHub. The difference, however, is that with these commands, we can put our cursor in this box and click the run button, and that will actually run these commands on this virtual server. So after putting the cursor here next to the exclamation point next to curl, I clicked the play button, and these commands were then executed on this virtual server. And this first step is installing Mamba Forge, which allows us to have the tools that we need in order to very easily install different analysis software on this virtual machine. So there's a number of steps, and this does take a minute or two to have it installed. So it's lots and lots of output as it's installing that. So now we are at this point to the next block of code in the notebook. And again, we're going to put our cursor in the block and press the play button. And as it's running, it is installing, first it's installing Trimmatic, then it will install FastQC, MultiQC, and Salmon. So I'm going to pause the recording and wait until that's done. So after a couple of minutes, it finished with the installation of all the software that we need. Then step 2 is to set up the environment, and it's just setting up a few directories on the server. And so I'm going to put my cursor there and run. As this is running, on the left-hand side, if we click the refresh button, we can see the directory structure. And so these make directory commands, we're creating a directory called data. That directory is right here. I can double click that to navigate inside. And then it's made a raw, FASTQ, trimmed, FASTQC, and reference directories. And those are right here. Now, there's nothing in these directories. And the first step 3 is to copy the FASTQ files. In this case, what I've done is to take just 50,000 reads out of the several million reads for each sample and make a FASTQ file that again is just 50,000 reads. This is something that makes the data set very small, and the tools then can run very quickly. I've placed these files that I created on what's called a Google Cloud bucket, which is a cloud accessible storage system. And this bucket is like a hard drive, and you can control access. So it might be just files that you alone can access, or maybe a group of users can access, or in this case, anyone can access. So you can choose the access. And on GCP, one can access these Google Cloud buckets. Every bucket has a unique name, and this is the name that we happen to use. And one can then access these data using a URL. And the curl program makes it easy to retrieve data using a URL. So I'm putting my cursor in this block of code and clicking run, and it is now downloading those very small FASTQ files. Step 4 is where we're preparing the reference transcriptome files that are needed for this alignment tool called Salmon. So Salmon is a tool that's used to align reads to a transcriptome and then report the number of reads that are estimated to map to any of those transcripts. And it's a fairly common, we used alignment program for RNA sequencing data, but there are many others. So Salmon is just one that we chose with this particular workflow, but there are many others out there. If you have a study in mouse or human, you might use a tool like STAR or HiSat2 to do those alignments. But here we're using Salmon. No matter what alignment tool you use, you must create a set of indexed reference files. So your reference might be the entire transcriptome, as it is in this case. So the set of all transcripts from the M. cheloni genome, or it might be the entire human genome or the entire mouse genome. And so depending on what your reference is, you need to then create a set of index files that are specific to the program that you're using. So Salmon needs a set of index files that are unique to Salmon. HiSat2 or STAR would have their own set of index files. But you must have the reference sequence and those index files on the server where you are running the analysis tool. So on the Google Cloud storage bucket, I do have a copy of the transcriptome from M. cheloni. And just to make it easy to access, I put a copy on the storage bucket, and we're going to copy that using curl onto the analysis server here. There's a data file that's needed to run Trimomatic, which are the set of adapter sequences that are used as part of the Illumina TruSeq kits. And so we're going to copy that. And again, I put that on the storage bucket. to make it easy for us to access. Step 3 is where we're actually running Trim-O-Matic. Trim-O-Matic is a tool that's quite powerful. It will trim your input sequence files to remove any low quality sequence as well as any sequencing adapters. And generally speaking, it's good practice to trim your reads prior to analysis. So I'm going to go ahead and run this. But to have Trim-O-Matic run, we of course needed Trim-O-Matic to be installed on the analysis server. And that's what we're using Mamba and Mamba Forge to do. Trim-O-Matic has a set of arguments that must be declared when you run it. So I have paired end reads. I want to use 3 different threads, so essentially 3 different processors. And then this is the read1 file, this is the read2 file, and a number of output files are all specified. And then other arguments, like what were the adapters that were used to create the libraries and other parameters. So I'm going to just put my cursor here in this block of code and click run, and it's running Trim-O-Matic. Because I had 50,000 reads, this runs very, very quickly. So Trim-O-Matic is already done. On the full data set, it takes slightly longer, but Trim-O-Matic does run very quickly. Step 7 is to run fast QC, which stands for quality control. It's a fantastic package to look at things like sequence quality. So I'm going to put my cursor here and run this block. And fast QC is generating a number of different very useful reports. When this is done, here we'll examine quickly some of those reports. If we look to the left, there are these directories that were created, that we set up. And if I navigate inside the FastQC directory, this is where the output files from FastQC are being written. And FastQC does create an HTML file. So I'm going to click on this HTML file, open that up, and here it is. So this is showing me how many reads are left after trimming. It's looking at sequence quality. These are all very high quality reads, and a number of different reports that FastQC is generating. And one can inspect the other sample as well. There were more than 2 samples in this experiment, but here with this, in order to make this workflow run very quickly, we're only going to look at 2 samples. And in the full tutorial with the complete data set, again, it's analyzing all the samples with all the reads and doing things as you would normally. When you run FastQC, you're doing it on a per file basis. And multi-QC is a way of stitching those reports together. So multi-QC, when it's going to read in the FastQ output and create a new output file. Here, where it's generating the output, if I go back up to the data and then back up to the RNA-Seq notebook page, this is where the multi-QC report file is located after it's done running. And I can double click here and view the multi-QC report. So that's accessible there. So if I go back to the notebook, the next step is indexing the transcriptome. And now that we have our index transcriptome that's still running, there it is, we can then finally map the reads for the 2 different samples. Here we're mapping the read ones in this experiment, just the read one files, and mapping them to our transcriptome index that we just created here. So this name here maps what's being created as the indexer is running. So we had that same name in this argument right here. when we generated the index. So this is the name of the index. So scrolling down to step 10, putting my cursor here and clicking run. So Salmon very rapidly is mapping the reads to the transcriptome. And again, this is running so quickly because we have so few reads. It's not the complete data set. And it's also doing it for just 2 samples, not the full data set. So all this is running. There's lots of what we call standard output that's generated. But importantly, when Salmon runs, if I go on the left here to the file menu, click on data, and the output of Salmon is in a directory called quants, short for quantifications, and expand that. Then we have these 2 directories, one for each sample, and the output from Salmon is here. And this tab-delimited text file called quant.sf is a file where the count data and other estimates of gene expression that Salmon generates are all listed. So step 11 is just giving us a sense of what the quant.sf file looks like with this miniature data set. And again, it's just the analysis of 50,000 reads, not the full data set. So what I'm doing is to sort the contents of this file by the 5th column. The 5th column has the number of reads. And then I'm just reporting the top 10. So I am put my cursor here and click run. And these are the top 10. Now, these read counts, this does say that it estimated 55 reads mapping to this transcript. That's not very many. But again, this was just by analyzing the 50,000 reads out of the millions and millions that were generated in the full data set. So that's why these numbers are so small. If I look at the other sample and look at its quantification file, we can see that some of these have, in this case, this first gene has 107 reads that were estimated to map to it. But you can get a sense of the data. In this published study that the Molloy lab had that was published in BMC Microbiology last year, there was one gene that they focused on, an acyl ACP desaturase. And so let's look at that. So there is a unique gene ID that is BB28 _RS16545. And we can look at that row of data from the quant file for this particular sample. So I'll run that. And so here in this miniature or subset of the data, there were only 4 reads that were estimated to map. If I look at the other sample, there were estimated to be 1 read. So again, very little, but this is because we were only analyzing 50,000 reads and not the millions and millions in the full data set. So this gives you a sense of how to run this particular workflow through. And at this point, there are a set of gene counts, and there's a workflow that one can follow to analyze a set of gene counts that are featured in the 3rd tutorial. And all of these analyses are done within R, and we chose to use the DESeq2 package to do that. After running all of the steps in this workflow, it is important to then shut down the virtual machine and so that it's no longer running. Because as it's running, you are accumulating charges. There's not a lot of money, but if you left your virtual machine on for several days, obviously it could cost quite a bit. And so it is important to do that. Before you shut down the machine, you may want to download data. And all of the files, of course, that we're generating, things like the quant files, like this file here and so on, are things that one would want to download. So there are different ways of exporting and essentially saving your files to your laptop. This file is, of course, the count data and is for one particular sample. So we can download and save the file, and so that is saved on your laptop. One would want to do that with the other quant file. Other reports could be downloaded, like the multi-QC and other things. You could download everything, whatever you feel is important to save. But after you've done that, then you go back to the GCP VertexAI workbench page. And this is our virtual machine that I called video demo that's running this notebook. And I'm going to stop this by clicking this box and then clicking the stop button. And this will then suspend this. And at any point, we can relaunch this after it's stopped. And if we were to do that, when we open it up, it would look like just like it did when we last ran it. With all of the steps that we had run, would they run, all of the output files we generated would be there. So, for instance, here I have a couple of notebooks that I could restart at any time, right? They have a stop symbol because they're currently stopped. And right now we're in the process of stopping the virtual machine that was running the notebook we just had. So now this is suspended. And again, we could, if we click on having still this checkbox checked and click start here, that would then allow us to have the notebook and be able to then click open Jupyter Lab and be in that environment that we were just in. Alternatively, at some point, you may want to delete this entirely and just remove it. And that would be just by having the checkbox for the items you want deleted and then click delete. And that will delete that item from your account. And so that's what I wanted to show. All of these, really the purpose again of these videos are to show you how to use the GCP environment to run these workflows. But hopefully you can also realize that the same notebooks that we have on GitHub can be used on AWS, the Amazon Web Services, or Microsoft Azure to run the same workflows. So thank you very much and see you in the next video.